So good afternoon. My name is Jingyou, and I'm a third-year PhD student at Harold Pimentel's lab at UCLA. And our lab mainly focuses on statistical inference for the deep mutational scanning screen. And today I'm going to talk about what is an invariant score, or like more about what should be in a good variance score estimation. So what I think would be a good score would have two properties. So the first one is that it should have low bias, and then it needs to have reliable uncertainty estimate. So I'm going to use this illustration a lot in this talk, so I just explain what it is. So on the top is your true distribution of your variance score estimate. And it's on one variant, right? Because we know there's a lot of biological variation, even though it is one variant. And the bottom one is the estimated one. And I use these black dots to represent the replicate level estimate. And you want your estimate to be the same as the truth. So to do and to ensure a good ranking of the variant effect. And the second is that you want the reliable uncertainty estimate, because essentially you want to identify these loss of function gain of function variants, right? And to do that, we need to do statistical hypothesis testing. And if we don't have a good estimate on your uncertainty, it's really hard for us, like statistically speaking, what is the difference between this loss of function variant and Y type, right? So a good estimate should be able us to detect the loss of function gain of function variance. And in this talk, I'm also going to talk about a lot of sorts of variation in the variance score, both biological and technical. And hope you will enjoy my talk. Okay, so this is not a data analysis pipeline talk. If you want like a step-by-step package level instruction on how to analyze your DMS data, please check out Dr. Rubin's talk 4 years ago on YouTube. So there's 3 points I'm going to go through. First one is, why is statistical inference important in DMS analysis? Okay. So even though your deep mutational scanning might be like really complicated, from the perspective of a computational biologist, it only has 4 steps. Right? The step 1 is to generate the variant library. I don't know how you generate library, but you generate some library. And then step 2, you introduce your variant library into the host, right? Whether it's cells, bacteria, virus, or yeast. And then you do some selection based on the phenotype of interest. Finally, you do the sequencing. And then you send your FASTQ files to us, and we analyze the sequencing reads. Okay. Goal of deep mutational scans, we want to find a mapping between the sequence and the function. And people usually think it's like a function like one-to-one mapping, right? Especially in like the variant effect predictor, there's like one sequence and you get the protein function, right? But the reality in the DMS is that we are actually measuring some sort of fuzzy effects that is not going to be a function. So the first source of variation in your deep mutational scanning is biological variance, and we actually want to capture that in our variance score estimate. So we like to view this protein function more like a random variable, or it has a distribution instead of a fixed constant. Right? So for example, here we have a bunch of cells. We have Y-type cells and variant cells. And if we, in the deep mutational scans, we do it as a pooled screen. So we don't actually measure the phenotype in the cell-to-cell level. But just imagine that we can achieve the single-cell resolution. You're going to observe the phenotype as some distribution, whether it's normal, it's a Poisson, negative binomial, or even there's double peaks, I don't know, right? But that's something eventually you want to learn at the single-cell resolution level. And the second source of variation in deep mutational scans, which are technical variants, is something we don't want, right? From step 1 to step 3 is highly experiment dependent, and I'm not going to comment on this because you all know experiments the best. But for the sequencing step, there's like been years of like statistician and computational biologists like study what would be the outcome, what would be the technical variation in the sequencing step. Right. So random sampling sequencing step is a major source of technical variation. So if you think of you have a pool of cell count and you do experiment like some DNA extraction, amplification sequencing to get a sequencing read, and you send the sequencing read to us, we can do the variant calling to get a sequencing count. So for the first step, there's been a lot of RNA-seq folks that already proved that the sequencing replicates can be captured using a Poisson model. And if you're unfamiliar with Poisson distribution, here is a little figure of Poisson distribution, and it's actually a very noisy distribution, in my opinion, because just look at this lambda equals 6, which is mean equals 6. It's like have a wide range from 0 count to 12 counts. Right. And adding on to that, we have the variant calling step. And there's an interesting paper published last year called SAM-Utils that benchmark different variant calling methods, including the popular one like Enrich2, DMS tools, DIM-SUM. And they all achieve like different precision recall. Right? Right? Aggregate like all sorts of variation in DMS, you're going to get a noisy, like somewhat noisy estimate for sure. So here's some like replicate notation adapted from the DIM-SUM paper. So first we have the variant library, and then we introduce the same variant library into your host, and that will become experiment replicate. And after you have this pool of cells before the selection, you do like a couple of rounds of selection, and that's selection replicate. And after you get the cells after selection, you'll do the sequencing a lot of rounds, and I call it sequencing replicate. I just show you that the sequencing replicate would be a more over dispersed version of Poisson. Right? And adding up like the other technical variations in all your steps and your biological variation, right? The DMS statistical inference is actually pretty hard. And well, what makes it worse, right? Like effect estimation DMS is really hard due to small sample sizes. Like I've seen a lot of experiment papers just to do like 1, 2, 3 replicates or like 1 selection rounds. Right? And I'm going to show you a little anime. Oh wait, I think this one got, yeah. But if you have 3 sample size, you cannot even see where is your variance estimate, right? You cannot even see the bar. Only after like 20 sample size or 30, like by the law of large number, you are going to have a good uncertainty estimate. And a good uncertainty estimate is very important when you do hypothesis testing to identify which variants are actually loss of function or gain of function or different from your Y type. Right? And here I'm trying to show you why it's a problem. The naive estimate lacks power to detect these variants. So for example, here we have a loss of function variance. And in the ideal case, you want to distinguish that this blue distribution is indeed different from the orange. Right? A correct label estimate would be something like this green one that we can successfully identify it as loss of function using like the, I don't know, like p-value threshold 0.05, sorry. Okay. But the naive estimate, because you have like an over-dispersed of the count estimate, your uncertainty is very wide. So even your loss of function variant might be falsely identified as neutral. And that is not something we want, or we say it lacks power. So like the solution is to incorporate more assumption into the modeling process. And there are some amazing papers published earlier. For example, Enrich2, it computes uncertainty estimate assuming concept Poisson distributed. But we mentioned that just by the sequencing step, you can introduce a more than Poisson distribution. So DIM-SUM, they write a paper in 2020 saying that, okay, so after aggregating all the technical variance, count variance is definitely more than Poisson. So they use another like target function to learn the replicate level variance and try to do a better job at the inference. Right, and to show you why, just For example, using a Poisson distribution, overly strong assumption would be a problem. So for example, here, we have a neutral variant now. So we want to identify this variant is not statistically significant different from this Y type. Right? A correct effect label will give you a p-value larger than 0.05, but an estimate with this one, for example, an overly strong assumptions like a Poisson will underestimate your uncertainty or technical variation and would be all biological variation, and it would be identified as loss of function. And that would be a false discovery rate, something we don't want. Okay. So hope this is my introduction section. Hope I convinced you that statistical inference is important in DMS analysis and why you should not just use a naive method to compute your log fold change. Okay. And the second part, I'm going to convey some really simple messages that Harold initially wants to be here to convey. So how can we reduce technical variations through better experiment design? Okay. I'm going to show you a lot of simulations here. These are simulated under the best case scenario with only sequencing variation included. Harold really wants me stress that this is the best case scenario. Okay. And the analysis is using just the most naive method. It's either linear regression or log fold change. And just know that this result applies to every variant. Even I'm only showing simulation of single variant, this result applies to every variant in the experiment, and that can be tens of thousands of variants in a mutational scanning screens. Okay. The first scenario, it cannot be simpler, right? Unstable statistics if you have low counts. For example, we have two experiments. One, this blue variant has 100 cells before selection, and the second variant only has 6, right? And we do some perfect selection, no selection bias, hopefully we can select 50% of the cells, right? And after selection count would be 50 and 30. And then we do we try to simulate the sequence instead of using Poisson distribution 1,000 times and see what will happen, right? In your first experiment, everything looks great. The green bar shows 0.5, exactly what you want to estimate, right? There's some variation around 0.5, but I think it's okay if you do multiple replicates and it's fine, right? But for the second experiment, you want to have 6 cells, and the chances of completely misclassifying this variant is 10%, right? More than 10%. And this is completely misclassifying, meaning that you are showing that your cells after selection is more than the cells before selection, and you clearly know this is wrong. Right? It's 10%. It applies to every variant with low count. Okay. The second simulation is trying to show that the dropout could confuse this ranking. Yeah. This is also very intuitive. Now we have 3 variants. They all have near perfect coverage before the selection cell count. And then assume that this is a growth screen. So the true cell doubling rate for variant 1 be 5, variant 2 is negative 5, and variant 3 is negative 10. So we assume that the variant 2 and 3 are both loss of function. But we do know that the variant 3 is more loss of function than variant 2. And the goal here is trying to rank variant 1, 2, and 3. And now it becomes a problem, right? Because, this is the theoretical expected cell count after the selection, right? You have a lot of variant 1, you have 3 variant 2, and you have 1 or 0 variant 3, depending on your chance. And chance of misranking variant 1 and 2 is 0%. Right? It's good. You are going to show that your variant 1 is I don't know, some gain of function variance of neutral. But the chance of misranking variant 2 and 3 is as high as 40%. If you just do coin tossing, it's 50%. I just let you know, right? Now your chance of misranking variance is 38.4% by this round of simulation. And also, that applies to every pair of 2 variants with low count after the selection. So, yeah, it's pretty bad. And I just want to make another note here, because I know there's also computational people in the audience, and we build a lot of variant effect predictor. And it usually either uses like clean var data to compute like the AUC curve, and or they use DMS variant ranking as the gold standard. So they compute the Spearman correlation between your DMS experiments and the variant effect predictor and hope it's like 100%, right? But you see, like only some of the best predictors, like I took out this like TPK1 gene. I don't know what is this experiment, right? So the best predictor is like the ESM2V, and the Spearman correlation here is only like 30-something%. Right? And if I'm a computation people, I see this result. Okay, the first thing I say that okay, ESM is really good, right? The second is that okay, so why is only 30-something%? Right? It can be due to experiments, right? Or it can be due to my variant effect predictor. But why there's a problem, I actually don't know. So yeah, just to keep in mind that even your experiments is not targeted to rank like the most loss of function variance or most gain of function variance. Like it will have an effect on like the other metrics you use to analyze this variant effect predictor. Okay, so my message one is so simple, right? Just to get a high variant coverage in the library before and after selection. And honestly, I don't know how to do that, but yeah, just try hard, okay? Yeah, if you're, and also like, know that if you're doing double triple mutation experiments, this rules applies to every combination mutation, right? And that space will be huge if you're doing double, like 500 amino acid positions with like times 20, and then you have to do like an exponential scale. So I don't know, right? It's hard. Okay, and the second magic, like the third simulation I'm doing, like low counts how replicates can come to rescue. So again, we are having this like really hard like difficult blue variant that just have 6 cells before the selection. And I'm here just trying to show that if you only have one replicator, right, just one, that happens to have a good coverage on this blue variant before the selection, right? Still the same, assume perfect selection 50%, and after selection cell count, your chance of misclassifying this variant drops from 15% to 5%, which I think is pretty significant. Yeah, I don't know. Yeah, so, and just to stress again, this is the best case scenario. We're only considering the sequencing variation, right? There's no biological variation. There's no other technical variation from your like library and like from like intra-transfection from like selection. So my second message is I need you to do like more replicates. If you're doing one, like think about 2, like depending on the budget. If you're doing 2, I think like 3 is a pretty sweet spot. If you can do like more, that's of course perfect, right? Just doing one sometimes like make our job too hard. Okay, and now I'm going to like do the third part. Your after experiments done, like just for example, you only do 1 replicate. That's your budget, right? How can we improve the variance score estimate? Right. I just showed in the introduction section that you can incorporate a lot of experimental assumptions, such as like count distribution, into a modeling process, so it will help to stabilize uncertainty estimate, right? And here I'm going to focus on biological assumptions because we just we just have a paper get accepted using biological assumptions, and we show that how we can. Hopefully, we show that biological assumptions really help improve your variance score estimate. Okay, so when I was first introduced to the deep mutational scanning like a year and a half ago, like by the Willow, he showed me this heat map, right? And I just like eyeballing this heat map like for a long time and realized there are like some structures like in the deep mutational scans. Right? The first observation is that the mutations at a position are correlated, right? A position could have like completely neutral variant, or it can be like complete loss of function or gain of function. Right. And the second is that despite being correlated, some substitutions behave differently. I think like intuitively, like, even though I don't know much about biology, it makes sense because different amino acid has different properties, and depending on what position it is, right? So, like for example, the amino acid substitution, like if it's like really like similar to each other, like with similar charges, like you expect the substitution to have like a more neutral effect. If it's different, it might be like loss of function. Right, and the scale of the substitution, in fact, each position is different. Yeah, I mean, yeah, of course, it depends on the position. Right. So we developed this method, new method called Rosas. It assumes mutations within the same position correlated, which is our first assumption. I was doing some proofing of this manuscript this morning, so it got accepted, and we have this GitHub link. It's built as an R package. I know a lot of folks using Python, but it's an R package. Yeah. Yeah, I mean, University does a like good job like building GUI stuff. Maybe it's something we want to do later, but yeah, currently it's here. So just using like a more mathematical term to describe how we build this assumption into our model. So the prior, we use Bayesian method, so we believe in the prior. So, so the prior of your variance score is going to be a normal distribution on your position score and position variance. And these are also estimated by the data. Okay, I know, like, I don't know how familiar, like, with Bayesian method like you are, so I'm going to give you like a little tutorial analogy of what I think is Bayesian method. Okay, so Bayes is a tug of war, right? So it's a tug of war between your prior and your observation. And the green part is your Bayes estimate. So when the number of observations is larger, your observation is given more weight, so the prior influence are weaker. But if you only have one replicate, think about only 2 data points, like the prior will have play a bigger role in your final effect estimate. So yeah, just like in some like informal term, I would characterize this beta Bayes to be some proportion of your prior and some proportion of your data, right? And if the data is like observations like more than 20 or 30, you'll see that how data completely overpowering your prior. And my method, it's like more complicated than a simple like Bayes, but it's a tug of war between your position and the data. So now our results estimate will be a tug of war between the position mean, which is also estimated data. So it has a little orange figure even under this pink figure that are the observed functional scores at the same position, right? And there's like the blue figures that are your true observed functional score or your MLE estimate, right? So there's a balance, like you want to find the sweet spot that, okay, so you want to reduce as much as outlier as possible by using the position mean as the prior. But you don't want it to be overpowering. I think that's the essence of Bayesian, like how you can strike the balance between the two. Okay, and yeah, hopefully in the paper we show that it can lower false discovery rate while maintaining high power. Yeah, so that's observation 1. And I've been asking a lot, right? Like why you don't use amino acid substitution, right? Observation 2 and 3. And the short answer is really hard, but now I think I find a way to like include like the amino acid substitution. Okay, so these are some published work. So now we decompose the prior into a position score and amino acid substitution score. And we add another term called position amino acid sensitivity scaling because we realize not every position has the same, amino acid substitution does not affect the variant the same fashion for different positions. Right? There's a lot of technical details behind it, so if you're interested, check on my post at 17. Yeah. And just think about it. If we are going to model like all the amino acid substitutions, that would be like 20 times 21 if you just consider single mutation. And in our case, we just consider single mutation DMS for now for this method. Right, that would be 20 times 21. It'd be 400 effects, and and if your like DMS only have 2,000 variants, right? You only got like four observation on your like amino acid substitution effect, and that would be also very hard, right? And we think that BLOSSOM provides a natural interpretation of amino acid substitution prior grouping for modeling. And something good about this BLOSSOM score is that it's simple, right? It only have like 8 or like 9 different levels, and we have clear expectation of how it will behave. If we have a more negative BLOSSOM score, we're going to see a more loss of function effect. And that's like a sanity check for us whether this model is using something correctly. Okay. This slide is in a rush, so hopefully I could explain what it means, right? Because we also estimate these like prior parameters, which is the functional score in the pink, amino acid substitution effect in the blue, and this amino acid sensitivity scaling on each position. We can actually plot the correlation between the position score and the amino acid sensitivity scaling. And if we just look at this plot, hopefully it could give us some insights into this protein and the protein function. So for example, we have the left-right one, which is where position dominates the effect, and there's like no amino acid substitution effect, so the scaling, there's nothing. It's a base method, so it just stays around like 0.5, yeah. And you will see like a wide range of position score. Or you can have the exact opposite, which is it has mild position effect, but amino acid substitution dominates, as shown in the most right figure, right? And you see a wide range of scaling. And I think most protein data I see is like in the middle part, where like position dominates your gain of function, loss of function sites, where amino acid substitution effects dominates at neutral site, and you also have a wide range of scaling. I've been seeing data for like the first 2 scenarios, but not really the third one. I'm not a biophysical chemist, so I could not tell whether it's something reasonable, like theoretically, to assume or not. So if you have any like feedback on this, please talk to me. I appreciate all the discussions. Right. And we apply this method. Just show you an example on the ARC1 data and how it shows like again, this arch structure where like in the most loss of function or gain of function side, it has low amino acid sensitivity, while like in the middle, it's like more amino acid substitution dominated. And just show you this is not some bogus effect, right? I point out some regions on the protein, even though I don't know what it is, right? It's some like transmembrane, right? So first is this red box, which is like mild position effect, but like high amino acid sensitivity effect. And you can see that you do identify these positions that since you have a wider range of like mutation effects. Right. Also, in the same region, you can like see this like blue box that your positions now completely loss of function. Right. And there's also sites that are like mostly gain of function. Right? So like these are examples just to show you that okay, so like it's highly possible that my method works. Okay. So, conclusion, hopefully I convinced you that statistical inference is important in DMS analysis. Just like pick your favorite DMS tool to use, not use some naive method. Hopefully it's Rosas, but yeah, I mean, it's too much, I know. Right? And how can we reduce technical variations through better experiment design? So 2 simple messages again, high variant coverage, and more replicates. And after experiments done, how can we improve the variance score estimate? You just by including more assumptions, like just find the sweet spot of assumptions. Like for example, Rosas incorporate biological assumptions. And we also have this new method build. Now just name it as Rosas BLOSSOM and check my poster. And we also develop a facts-based like a method specific. These are for the growth, like rosaceae and rose blossom. These are for the growth screen or binding screens. And we just developed a new method that's specifically on FACS screen. So 4 bins. You can not only, now you can like see it as a distribution change instead of like computing some like average count of the bins. Right? We are preparing that manuscript, and hopefully it can also build as a Python package or R package. Okay, finally, I would like to thank my lab, especially my PI, Harold. He's supposed to stand in here, not me. I was supposed to give a research talk, but yeah, due to some like unfortunate medical conditions, he could not travel this week. And we have Jerome. We've been collaborating on the fact screen. And also, big thanks to Willow and Matt, Chris, like members in his lab, and James Fraser. Yeah, and yeah. Please give Rolas a try. Thank you so much. I'm happy to take any questions.
Thanks, Jingyou, for a very nice talk. Maybe to start, I could ask, you talk about the value of more replicates as your recommendation to deal with, I guess, the stochasticity of low variant counts. Have you thought about, you know, if you had a limited sequencing budget, the value of adding more time points versus just the sort of before versus after selection in these kinds of assays? Do you get, you know, more information by adding time points?
Yeah, I know all the Willows data have 3 selections, so it's like 4 time points. So I kind of know the value of like if you're doing more selection, of course it's better, right? But like the simulation here, I'm just trying to show like how variant coverage is important, because even though if you do like multiple rounds of selection, if you have like low variant coverage at your first time point, this variant has a high chance you get like screwed up by the variance effect, like inference. So yeah, selection, like more selection round would also help.
Hey, thanks for the talk. It's really interesting. I have a couple questions. If you can't answer them, I guess we can discuss later. But one is about your Bayesian method. Super interesting. I was wondering, like, would you recommend not using the raw DMS data and using the score from your algorithm? And in that case, like, how do you select the prior? And do you feel like the score, like, I don't know if this is something you plan to do, is that the score that you get from your algorithm would be like more correlated with ClinVar than the raw data? And then number 2 is about the experimental assumption, for example, the counts. You mentioned that if you don't do any assumption, naive estimate would be underpowered, and if you have overly strong assumption, then you would like have an kind of false precision when something should be neutral. So do you have a recommendation on how to handle that? Looks like you showed those 2 papers that are somewhat different.
Okay, there are a lot of questions. Okay, well, what's the first one again?
Yeah, like, would you recommend using your Bayesian methods rather than raw DMS score? And is that, do you feel like that would be more correlated with ClinVar?
Yeah,
and also how to handle the prior.
I mean, that's my message, right? You should use a proper DMS tools instead of like your naive estimate, if you call that your raw DMS score. Yeah, and the second is that how you choose your prior, right? Like here, because like if you're using empirical base, you need to choose your prior. But if we are using a full Bayesian method, right, using like some samplers, so actually your prior is also estimating your data at the same time. So that's why, like in the Blossom project, we show that okay, you could use like the estimation of the prior to actually inform you something about the proteins. Yeah.
Hi, great talk. I'm Kyle from Cold Spring Harbor, and intrinsically disordered regions there's some evidence that for them, amino acid composition dominates over the exact order of the sequence to determine function. So have you tried running your scaling model on any intrinsically disordered regions to see if they might fall in that third scenario?
Oh, no, I haven't. Thank you for mentioning that. We could talk about like the data set later. Yeah, thank you so much.
Thank you.
Thank you for the really good talk. I really enjoyed that. It seems like kind of one of the key results from your work is that when we're in this regime of like low coverage of variants, then the explicit ranking of certain variants can be very unreliable. And this is maybe a bit of an overgeneralization, but I feel like a common outcome from DMS studies is you get this sort of bimodal distribution with a cluster of variants that have this like neutral or wild type of effect, and then a different cluster that is like loss of function. And I guess I'm curious, like, do you think with existing data we're well powered to do that question, like putting variants into those bins, but not necessarily explicitly ranking them within the bins, if that makes sense?
Yeah, I mean, like, I think a lot of goal of the DMS screen I see is to this kind of clustering, right? You want to cluster what is loss of function, like what is the pathogenic or what is neutral. Yeah, I think the point I'm making here is just like, okay, maybe your experiment in your paper, you just want to classify your variants. But like other people, like us, we don't generate data. So we're going to use your data to do some things that your data is not supposed to do, right? For example, the ranking. Yeah, so that might cause a problem. Right?
Thank you.
Hi, I'm Jason Commander from Mass Pioneer. Thanks for the great talk. I had a question about kind of how much you know how to trust the Bayesian models. Like, let's say you have this variant and it's going, your functional assay is going to go in a ClinVar, and somebody's either going to get a treatment or not because of it. So it's real important to know what the truth is about this variant. And say all the other 18 amino acid variants, they're all loss of function, and it's a lysine, and those tend to be loss of function. The model will like really almost just automatically say that that's loss of function. But we went through all the trouble to do this experiment, and it's almost like that data, in the extremes, not even used. So like a biologist might say, well, just do the naive model, and I'll put my assumptions into it later. So can you help kind of like convince the mindset that we should, trust that kind of Bayesian influence from one point to the other?
Yeah, I understand this. We actually got a lot of requests from reviewers to like actually show that even in one position, like our method will not overpower, like, because it's a tug of war, right? It's like how much your prior plays a role in your final estimate depends on how much data you have. And we show that even though in one position, like we could, okay, if you just compute the Pearson correlation between our score and naive score, enriched 2 score, like whatever method is, like you get like 95 or 99, like 95, 96% of correlation. So I would not worry too much about it. Yeah.
Thanks.
Hey, you mostly presented this as a tool for refinement, right? Have you ever thought about using it for imputation to predict from missing data instead?
I mean, it can, right? That's not the focus of like our method, but I think there's been imputation work using Bayesian method, and especially like some HM, or not HMC, like some like Markov model, like so yeah, I think so.
Okay, thanks.
Hi, I have 2 questions. One is, so you have this Bayesian prior that's either based on position or amino acid substitution. Do you think the next step is integrating more complex, like protein language models, priors into the methods?
I don't really think so, right? Because if we get a result using priors, like some like, for example, alpha missense or ESM, that would be, I think, be overpowering, like, because I mean, it's already there, right? It's not like, okay, here we build this prior as just like some like loose structure of your modeling. So it does not actually influence you very a lot. But if you're going to like have like very complicated method, like even like deep learning, it's divert from the initial point of our doing just the statistical inference on the data. And we care a lot about the experiment itself.
Okay. And did you look at, you show the problems of benchmarking fitness estimators. Did you look at the differences when you correct scores with your methods?
Not really.
Okay.
Yeah, but I talked to like Vasilis and like some other folks doing this. Like a while ago, and they all think that probably if we can like improve the DMS score effect estimation, like probably you will see a like Bayes-Milne correlation of their predictor and the data. Yeah.
Okay, one more.
That was a really fantastic talk, and the preprint is amazing. You know, there are lots of pernicious reasons why one could worry about the basic approach related both to the fact that the, you know, and this was asked before, like how much of a positional effect occurs, changes from position to position and protein to protein, but then also like the many vagaries of different assays, right, which will have different pathologies of what their score distributions and stuff look like. So have you applied this method to like many more data sets and kind of, I mean, it looks amazing, but if I understand the preprint right, it's kind of one data set, right?
I mean, like in the preprint, okay, in the preprint there's only 2, right? Oh, like 5, but requested by the reviewers with the like 6 more in like the official version, right? And we also used claim var data and alpha missense data and also EVE to benchmark against different methods, and hopefully that's enough.
Great, thank you, Jingyou.